Good day, everybody. Thank you for coming to our ZOOMinar series. Today we will have two speakers. The first one will be Dr. Gomes, and the second one will be Dr. Klenerman. And starting with Dr. Gomes, he's an associate professor and a coordinator of the protein misfolding and amyloids in biomedicine laboratory at the University of Lisbon. He received his PhD from the New University of Lisbon, and after a year of postdoctoral studies at Helsinki University, Dr. Gomes became a group leader at the New University of Lisbon, and later he joined the University of Lisbon. Dr. Gomes has been highly productive through his career with more than 120 peer-reviewed papers, 10 book chapters, and 3 books that he published. He is a biochemist whose research focuses on molecular mechanisms underlying protein misfolding and aggregation in neurodegenerative diseases. And his lab combines molecular, cellular, and biochemical experimental approaches to investigate protein structure and self-assembly in vitro and in cells to answer these questions. Thank you so much for coming today and just meeting you again. It's your turn now.
Thank you very much, Magda, for the introduction, and thank you and Hans and all the other organizers for the invitation to present our work. It's really a pleasure to participate in this very nice ZOOMinar series, and I congratulate you for the effort in putting this together, because it's really an outstanding collection. So in the next 30 minutes, I will overview what has been recent work in my laboratory. As Magda pointed out, we are based in Lisbon, Portugal, at University of Lisbon. And I would like to start by thanking my amazing group and collaborators that I will refer more specifically towards the end for this contribution and for their participation in our work. So my laboratory has a long-standing interest in understanding protein aggregation phenomena from a mechanistic viewpoint, but also we've done a lot of work in the past on how metal ions influence protein folding and aggregation. And we have the ambition to kind of recreate some of the complexity of the biochemical reactions that take place in the brain, in the extracellular synaptic milieu, in the test tube, by doing experiments that are cued by evidence that we find from the physiological data. And more recently, we got particularly interested on regulatory protein-protein interactions that interfere with these processes. And specifically, we got an interest on this protein called S100b, which is the focus of today's talk. So in a nutshell, today's story is about the chaperone activity of the S100b protein, which, as I will show you, is able to block protein aggregation, seeding, and mitigate its toxicity, and that we think is very relevant in Alzheimer's disease, as well as possibly in other neurodegenerative disorders. So introducing S100b, so this is a very abundant protein in our brain. It's about 0.5% of our total brain protein, but actually it's a member of a broader family, the S100 protein family. So these are small calcium binding proteins. They have 2 EF-end calcium binding sites. They have intra and extracellular functions, and many of their functions depend on the expression levels that can be either low or relatively high. They're also tuned by their oligomeric states, so they assume different functions depending on their oligomerization states. So as you will see in the next slide, they occur mostly as dimers, but they occur also as tetramers and hexamers and octamers. So actually it's a family that is composed by over 20 different members, which have tissue and cell specific expression, although they are structurally identical. And here I'm depicting the structure of S100b in the apo state depicted in orange and in the calcium bound state in blue. And it is obvious the substantial conformational changes that take place at the level of helix 4, which tilts by around 90 degrees upon calcium binding to one of these EF hand binding sites. So this conformational change that takes place upon calcium binding is important for some of the functions of S100 proteins, and it is certainly important, as we will see later on, for the activity of this protein as a chaperone. So it's a beautiful protein, as you can see, very stable, very well behaved. Now, its function, as I've mentioned, in the brain is dual. So under normal trophic conditions, it is essentially intracellular, and its extracellular levels are relatively low. So it is involved in these cases in trophic functions. But upon neurodegeneration, or upon aging, or upon traumatic brain injury or Alzheimer's disease, then the expression level of this protein increases, and it is secreted together with other alarmins, and they are all either called alarmins or danger, damage-associated molecular pattern factors. So under these conditions, the level of this protein extracellularly increases up to very high concentrations. Now, one important aspect that I want to draw your attention to is that S100 proteins are known to be multifunctional. So as you can see here, they are involved in a number of diverse intracellular functions. Cytoskeleton organization is one of them, which will be important for the topic I'll be discussing later on, but trafficking, DNA repair, S100b is a protein that interacts, for instance, with p53, calcium homeostasis, so a lot of intracellular functions, but they are also secreted, and when they are released, they are able to engage a number of receptors. TLR4 is one of them, but also RAGE, the receptor for advanced glycation and products. So they trigger intracellular signaling processes that then play roles in regulation and transcription. Then they can be secreted as cytokines or chemokines and play roles in differentiation, proliferation, and migration. And then many of these proteins, they are associated to metastatic processes, and some of them are markers, well-known markers in cancer. So in a nutshell, they play different functions, different multiple functions. And a few years ago, we looked into the literature and kind of overviewed in the multiple ways in which S100 proteins were implicated in Alzheimer's disease. And this is something we've reviewed a few years ago. And I want to start by highlighting an effect on the aggregation of A beta by recalling some of our previous studies that show that, so we decided to investigate if these proteins, which are abundant in the brain, S100b is one of them, but in our brains we have also S100a6 and S100a8, among others. So we wanted to correlate this with the formation of plaques in the AP23 mice. So what we can see here is that, in fact, there is evidence for colocalization of these proteins, which are produced by astrocytes, and they are secreted and accumulate in the same regions where the plaques are. So this prompted us to look specifically into S100b, and here you have the contour of an amyloid plaque in a 5 FAD mice brain slice. And what you can see here is that, in fact, you have multiple staining of S100b, suggesting that the protein is abundant in the same place where A beta is aggregating. So if we look into an in vitro fibrile, and if we do TEM immunogold labeling analysis, we see that in fact we have interactions of S100b with the fibrile bundles, but also with oligomeric species. So this prompted us to, back in 2017, look into the interaction between these two proteins, so amyloid beta 42 and the calcium-loaded S100b. So I'm go very quickly through this. This is work done in collaboration with Bernd Rift at the Technical University of Munich, but basically we've mapped the interaction site within a specific region of S100b, and we've shown that there is a physical interaction between A beta and S100b, and we have also demonstrating using kinetic analysis for A beta aggregation, which I will not go through today, that they actually influence primary nucleation, but also secondary nucleation events, because it binds also to fibrils and oligomers. So kinetic findings that we corroborated with TEM analysis. And of course, the interesting aspect was that S100b is able to restore viability and decrease apoptosis induced by A beta aggregates. So it plays a protective role that can be summarized in this generic slide, which kind of overviews data from a series of papers that we've been publishing on this model in the last years. So we had established that S100b acts like an inhibitor of A beta aggregation and proteotoxicity. We proposed the chaperone role. And when we looked into the literature and found evidence that, in fact, S100b is also there was also evidence, biochemical evidence for interactions between S100b and microtubules. Of course, this prompted us to investigate its effect or its possible interaction with tau. And this is what we did more recently, and we did it in multiple ways. So we started by using cell models to investigate if there was an interaction between S100b and tau in living cells. So for this, we have resorted to biomolecular fluorescence complementation assays. So in this type of assays, you have fluorescent protein split in two, and of course it's not fluorescent under these conditions, and you tag to it the protein that you want to investigate if they are interacting. And if these two proteins interact, they bring together the two fluorescent halves and you get fluorescence. So this is a schematic representation of what we get. In fact, we see that we can use this model to map tau into the microtubules. And then, of course, we did the same experiment, but in one of the constructs, we used S100b, so we would get fluorescence if there is an interaction in cells between tau and S100b. And in fact, this is what we get. So we could see that, in fact, in living cells, S100b interacts with tau within the microtubules. Now, of course, we wanted to perturb microtubules to kind of generate some destabilization similar to the one that occurs in pathology. So for that purpose, we used a drug called nocodazole, which disassembles microtubules. And when you disassemble the microtubule, of course, you lose the microtubule fingerprint on the image of the cell, but then you can wash it out and you can recover the microtubule network, and you still see some puncta, but if you wash out nocodazole, you restore microtubules. Now, we did this very same experiment, but in the presence of S100b. So we destabilized microtubules in the presence of S100b, and in these conditions, what we obtain is, in fact, an image or a set of images that shows that the microtubule network is not properly restored, suggesting that released tau is in fact kept busy interacting with S100b in the cytoplasm. So this prompted us to investigate if these two proteins were physically interacting, and for that we used structural NMR, a set of experiments that we did with Isabelle Landriot at Université Lille. And our first observation is that in the apple state, when calcium is not bound to S100b, we see that there is a very poor binding to tau. However, the scenario is drastically different when the same experiment is performed in the calcium-bound form of S100b. In these conditions, you see that there is a region in tau in which you have a strong binding. And if we map this into tau sequence, we see that this interaction is taking place at the microtubule binding region. So we've done this for the full-length tau protein, but we also did it for the K18 fragment, and we've done it to using smaller fragments that include the PHF6 domains, and I've identified that in fact these segments are interacting with the calcium-bound S100b protein. Now, when we map this on the protein structure, and here I have a color code in which red is the strong interaction, actually you see that binding occurs predominantly at this cleft, which is formed at the interface between the two dimers, which interestingly is exactly the same region which we have found to interact with amyloid beta. So this suggests that in fact this regulatory interfacial cleft is the region to which dynamic interactions take place between amyloidogenic clients and the S100b chaperone. Now, of course, we wanted to verify that this interaction, get some structural data on this interaction, and then we did SAX analysis. And what we get is a density. So we did this experiment with tau K18, and we got an additional tau density, which corresponds to tau K18. So this is the SAX analysis of the isolated complex obtained by size exclusion chromatography, and this is also in the paper, and these were experiments done by our collaborator and friend Günter Fritz, now at Hohenheim University. And by just for fun, we've more recently looked into how would the complex look like from the perspective of AlphaFold prediction. So we asked AlphaFold, what would the complex between these three peptides look like? And it's very exciting that you get a very similar structure. And in fact, here we have the PHF 6 regions which are binding to these two helices, and they are placed exactly in the same site for which we had obtained NMR evidence. So this shows kind of powerful tools that we structural biologists have today at our reach. Now, of course, the important question would be to find if S100b could influence tau aggregation. So we started by kinetic experiments using the full-length protein. In the absence of calcium, we see that there is almost no effect. So the endpoint, we see fibrils. But when we do this very same experiment in the presence of calcium, calcium-bound S100b binds strongly to the microtubule binding region, and we have a strong inhibition at substoichiometric levels, and you basically don't see fibrils being formed anymore. So we've recapitulated these experiments using the K18 fragment, and we got the same type of results, even more striking. So at very low molar ratios, we see a very striking inhibition of the aggregation of K18, which we then proceeded to a mechanistic analysis, which is detailed in this paper. Then we wanted to test what would be the effect of having S100b at different stages of the aggregation reaction. So here we have the aggregation of tau, and if we had S100b at the beginning of the reaction in the lag phase, basically we don't have any fibrillation going on, so suggesting that it's binding to monomers, as we have seen. And by the way, this gray line is the control with S100b, just to show that, of course, it's not fibrillating under these conditions. Now, when we allow tau aggregation to proceed roughly to the reaction half-time, and when we have S100b, then we see that again we block further progression, so suggesting it's binding to oligomers. And when we add it at the end point, although we see binding to fibrils, there is no reversal, so it's not able to redissolve the fibrils. But as you can see here, we have carried out immunogold labeling experiments in which at all these different stages, and we see binding of S100b to fibrils, to the side of fibrils, accounting for effects on secondary nucleation, but also to other types of oligomers. So the proteins interact, the proteins, the S100b has an effect, delays aggregation. Does it have an effect on toxicity? And for this, we tested what would be the effect of S100b on a model of cell-to-cell spreading. So we used this tau biosensor Fred cell model. So this is a model which is very well established in the literature. Basically, if you expose these cells to tau oligomers, then tau oligomers are uptake and they will see the aggregation, and you measure intracellular puncta tau aggregates. And this is what you get when you look at the images, and you can quantify, and you see that you have an effect which is proportional to the amount of tau oligomers, and this is basically the control. Now, these are experiments that we did with Cascade at UTMB in Texas. And then, of course, we did this control experiment as a primer to check the effects of S100b. So when we do this experiment in the presence of S100b, we see that actually this seeding effect is blocked. We get much lower amounts of intracellular tau aggregates being formed in these biosensor models, showing that S100b is able to block this pathological transfer. So overall, S100b prevents tau aggregation and seeding from cell to cell. And more recently, we've looked into possible effects on the formation of liquid droplets by tau. So you know that tau forms liquid droplets under high crowding and high concentrations. And this is the kind of phase diagram that you get. And this is relevant because tau LLPS can initiate tau aggregation, and they are hypothesized to induce pathogenic tau conformation. So we decided to test the effect of S100b on this protein, on this phenomena. So what we see is that actually when we compare the phase diagram in the absence and in the presence of S100b, we see that we need a lot more of Peck and higher concentrations of tau to get LLPS formation compatible with this sequestration effect. And once we have droplets formed, when we have S100b, then we see a decrease in turbidity, showing that in this case, S100b, the interactions with tau in droplets is able to be recovered partially by this chaperone. And of course, then we looked into droplet dynamics. So for that, we used labeled tau, and if we count the droplets under the microscope, we see that S100b has also an inhibitory effect, identical to the one we get when we look into light scattering, and eventually it may have some effect on dynamics, which is yet to be clarified. Now, so take-home message, S100b is this alarmin produced by astrocytes that is able to inhibit the aggregation of A beta that you have evidence of its co-localization with plaques. It is also able to interact with tau in microtubules and prevent its pathological self-assembly, to block LLPS formation and seeding effect. And we framed this evidence in the context of the progression of Alzheimer's disease by putting forward the hypothesis that, in fact, S100b, which is a multifunctional protein, as I've pointed out in the beginning of this talk, actually combines protective and disease-aggravating functions depending on the disease stage. So today we know that the so-called early inflammation plays a likely important role in early inflammation. At this point, you presumably have already increasing amounts of alarmins, including S100b, and yet you still don't have yet dementia or cognitive decline. So we hypothesize that under these conditions, S100b plays these protective functions that we've just described as a chaperone. But then as disease progresses and as the proteotoxic insult increases with the accumulation of A beta and tau aggregates, then this protective function is abolished and is reverted to, and the protein reverts its action to its pro-inflammatory activity. Then it will exacerbate gliosis and then basically be a pro-inflammatory molecule. So take-home messages: S100b undergoes regulatory interactions with A beta and tau, prevents aggregation and toxicity of amyloid beta and tau. It is a novel type of an extracellular chaperone from the alarmin family and is a potential druggable target because now we are working on ways to fine-tune this chaperone activity towards specific species. And last but not the least, I want to thank all the people involved. So this work has been spearheaded by Guilherme Moreira, first-year PhD student in my lab, extremely talented, but we counted with very important collaboration with colleagues a little bit around all over that are kindly collaborating with us and providing us with their expertise. And I thank them, and I thank all the other co-workers involved in other aspects of our research that I did not mention today. And I thank you for your attention. I'll be glad to take any questions you may have. Thank you.
Thank you so much, Claudio. That was really great talk. We already got questions that's in the chat, and I'm going to read it for you. This is coming from Rams. He says, great talk, Claudio. Thanks very much. Do your data comparing the difference between the binding affinities for A beta monomers, oligomers, etc., with S100? I guess he meant, do you have? Can S100b de-oligomerize to form monomers of amyloid beta? So let's first answer that question and then we'll go on.
Okay. So the binding affinity between and I take this promo in the meantime. So the binding affinity between S100b and A beta is relatively high, is about 1 micromolar, but we don't expect a very high binding affinity because these interactions are dynamic and the two proteins exist at very high levels. So we have no evidence regarding Ram's question on change of oligomeric state of S100b. If I'm getting the question right, we don't have any evidence. We have a very stable dimer and the protein stays as a dimer upon the interaction. So, and there's not even co-aggregation when this interaction takes place. So they form a stable transient complex. So that's the point. Now, for the second question, yeah, go on.
Yeah, Claudio, do you mind if I read it?
No, not at all. Go ahead.
Yeah, the second question, how does this effect vary with pH, salt ions, and in the presence of other cofactors, including
That's a very good question. We have not yet looked into the effect of lipids, but we have looked a little bit on the effects of metal binding to S100b, because S100 binds, apart from calcium, it has an additional binding site for copper and zinc. So it's a promiscuous binding site. And what we have seen, we have a paper on that, is that the protein is actually acts as a dual mode chaperone, because on the one hand, it is able to block and prevent the aggregation of A beta, but it's also able to bind excess of zinc. And you know that zinc induces the formation of toxic oligomers of A beta. So it has this dual protective function. Yeah.
So the third question is kind of going this way from Rams. Does zinc bound S100b do similar activity to the calcium binding?
Yes, it does. It does. So zinc binding does not impair this chaperone activity, and actually, in the context of, again, recreating some of the biochemistry of Alzheimer's in the test tube, when we know that we have elevated levels of transition metals, zinc included, we see that it also binds and plays a role. In fact, we have two papers on that, one on in vitro approaches and the other in collaboration with Andres Grabrucker, in which we've checked effects on neurons in primary cultures.
So you have another question. They're all kind of popping up in the chats, but after that I'll switch to the Q&A.
Okay.
It's coming from Simone Atanasio. Great talk.
Mm-hmm.
Do you have evidence for direct interaction between S100b and microtubules? And the second one, do you know if S100b is also attaching other IDPs like Synuclein? Actually, it's kind of my question.
Yes. So starting for the interactions, for other proteins from this family, there are studies reporting some interactions with alpha-synuclein, but so the answer is yes, and that would not be surprising, because I think that actually the reason why this chaperone acts as an inhibitor of aggregation may have to, or likely has to do, we hypothesize it has to do with the fact that these two helices at this binding cleft, they have some aggregation prone segments that promote possibly heterotypic interactions that delay the aggregation. So this could be a general aspect. Now, regarding the first question for the direct interaction between S100b and microtubules, yes. I mean, I went too fast, but I kind of show that with the BFC experiments that you can refer to in the paper.
So the next one is from Q&A Zoom. It's coming from Saeed Hadi. Thanks for your presentation. Did you compare the tau dynamics in the presence or absence S100b protein by NMR, for example? I think that you answered that, but yes, I give it to you.
So the question is if we compare the dynamics of tau
dynamics
when it,
in the presence or absence of S100b protein by NMR.
Yes, I mean, so for the interactions, what we did, again, in collaboration with Isabelle Andriot, which is the NMR expert on tau, was the interaction between the full-length protein, the K18, and different fragments. So, of course, we can map interactions into a specific region, but of course the protein is very dynamic, and we can clearly pinpoint interactions on the microtubule binding region. The other regions are just more flexible, in agreement with the sex structure that we got.
Next question is coming from Ashutosh. Very nice talk. Does Peck molecular weight play a role in LLPS?
Yeah, I refer this question to the literature on LLPS. So basically, we were just following what has been reported for the crowding conditions that promote the formation of tau LLPS. So I think so, but we're not the ones doing this. So there are lots of few excellent papers out there that report this.
The next one is from Murugesan. How does S100b release? I guess how does it's released into extracellular plate?
Yes, there are multiple mechanisms. It's not clear, but passive diffusion and it's one mechanism through secretion pathways which are not fully clarified, but it gets out there.
So there's another question from Ashutosh. In data where you show tau and tau S100b interaction that shows fluorescence, can the interaction be mediated by the fluorescent molecules carboxyl and N-termini interactions alone?
Yeah, this is a very good question, which points to a usual criticism for BFC experiments, that in fact you get this interaction because the two halves really want to meet. And of course, we did all the controls on Western blots using different cell models to verify this interaction. So it's independent of the fluorescent halves.
The next one is a third question from Simon Atanasio. Experiments you show are with nocodazole. Have you ever thought to size the cytoskeleton and see what happens, for example, with EPO-D?
No, actually, we have not. We have not. So when we did these experiments with nocodazole, the idea would be to destabilize the microtubules to see if we get a transient release of tau and what could be the role of S100b in the process. Because of course the underlying hypothesis to our observations is that S100b has these housekeeping functions in microtubule maintenance under normal physiological conditions. So it's one of its chaperone-like roles, but we have not tried this. Think about it. Thanks for the suggestion.
And the next one is from again Muru Gezian. Great talk. Is there any sequence difference between the intracellular versus extracellular S100b? No,
not that we are aware. It's the same protein, and in fact, we can even expose cells to purified S100b, and it gets inside cells and has effects. So this refers to one of the papers I was telling before. So to our knowledge, there is no difference. It's the same protein.
Thank you so much. I guess we will be moving to our second speaker.
Thank you.
If you can please stay, because we will have
of course
a Q&A session after that and with the discussion. While I'm introducing this, Dr. Klenerman, can you please upload your slides? And it is my pleasure to introduce the second speaker for our series, Dr. David Klenerman. He is a professor at the University of Cambridge, UK. He is a physical chemist who graduated and completed his doctorate at Cambridge University, working with Professor Jan Smit on infrared chemiluminescence. He did his postdoctoral research with Professor Digzer at Stanford University, California, working on high overtone chemistry. He then returned to the UK and worked for 7 years for BP Research in their laser spectroscopy group before returning to the Department of Chemistry, University of Cambridge. He's currently a Royal Society Glaxo Wellcome Professor of Molecular Medicine. He's a fellow of the Royal Society and the Academy of Medical Sciences. At Cambridge, Dr. Klenerman's work focuses on developing and applying physical methods, particularly laser spectroscopy and single molecule fluorescence to biological and biomedical problems. Thank you so much for coming today, and looking forward to your talk. Nice meeting you.
Well, thank you very much. So I thank the organizers for giving me the opportunity to present our work. So I'm going to talk to you about characterizing protein aggregates in neurodegenerative disease. So I'm going to start off by giving you a brief overview of the idea behind single molecule fluorescence and super resolution microscopy. And I'm going to show you how we've developed and applied these methods to study protein aggregation and neurodegeneration, with examples of work we've done on cerebral spinal fluid. And the main emphasis of the talk is to show you how new physical methods can enable new types of science. So the idea behind single molecule fluorescence is shown in this slide. You can excite a fluorophore from its ground state to its excited state, and then it can go back from its excited state to its ground state by fluorescing and emitting a photon. But if you have a fluorophore, you can undergo this cycle again and again and again. Unfortunately, you can't do it forever, but you can do it for about 1 million to 10 million cycles. So a single fluorophore is amplified and can give you 1 million to 10 million photons before it photobleaches, and you can easily detect these photons with a sensitive camera, and that's essentially the basis of how we can detect single molecules. As you're aware, there's a fundamental limit to the size of objects you can see with light, which is the diffraction limit, which is typically 200 nanometers. So this just shows you objects of different sizes, but low 200 nanometers, things like virus, proteins, or protein aggregates that we're interested in can't be observed, that they'll appear as a blob that's the size of 200 nanometers. But in the last decade, people have broken this diffraction limit. And one way to do it is as a stochastic method that I show here. So here I have a sample that's imaged, and you can see the features are blurred by diffraction. But I can organize things that I image a single molecule. My single molecule is still blurred by diffraction. It shows up as a blob that's 200 nanometers in diameter, but I know it's from a single molecule. So the single molecule must be at the center of this spot. And I can localize where the molecule is with greater precision, typically 20 or 30 nanometers. So I can replace my blurred spot with a spot with much higher localization precision, typically about 30 nanometers, and then repeat this process again and again and again to build up a super resolution image of my sample. In this way, you can break the diffraction limit. So this just shows you that we build home-built instruments. This is one of my postdocs, Alex Ponjovic, building one of our super resolution microscopes. And these typically have a bank of lasers of different colors, which are combined and come up an objective. Typically, we work in a so-called total internal reflection, where the light undergoes total internal reflection at the cover slip, but it undergoes an evanescent field is produced, so the light propagates a few hundred nanometers above the cover slip. So I can observe things in this 200 nanometers above the surface, and this removes a lot of background signal from things that lie about higher than 200 nanometers above the surface. In this example is a T cell, and I label the T cell receptor with an antibody. If I play the video, you can watch these receptors diffusing around on the surface of a live cell. So the focus of our work, as you're probably aware, is soluble proteins tau and A beta form aggregates during the development of Alzheimer's disease. And alpha-synuclein forms aggregates in the formation of Parkinson's disease. But these large inclusions, these large aggregates, are not thought to be the toxic species. It's the small soluble aggregates that are formed on the way to forming these large aggregates that are thought to be toxic. So we're interested in developing methods that determine the toxic aggregates and their mechanism of toxicity and find out the real species that cause the disease in humans. As you're aware, a large number of proteins aggregate in human disease, A beta in Alzheimer's disease, alpha-synuclein, transthyretin in familial amyloid polyneuropathy, prime protein in Crusher-Jacobs disease. And these are all characterized at the end by forming these beta sheet-rich filamentous aggregates with this characteristic cross-beta sheet, 10 nanometers in diameter, but very long. So it seems to be a very fundamental process. So what are the aggregates that cause Alzheimer's disease and Parkinson's disease? So this is not a cartoon. This is based on a careful study we did of the aggregation of alpha-synuclein. So you start with monomer, and it can form small dimers and trimers. And then we showed that this can undergo a structural conversion from these globular aggregates to these more fibrillar aggregates that can go on to form fibrils. And what we showed is that this conversion is slow. It takes 24 hours, and that these converted aggregates are much more toxic to the cell than these initial globular aggregates. But this slide also shows you the big problem we have. We have a range of aggregates of different sizes, dimers, all the way out to 100 mers, and we have aggregates of different structures, these globular aggregates and these fibrillar aggregates. So it's very difficult, unless we can resolve individual aggregates, to work out which aggregates are present and which aggregates correlate with increased toxicity. So for those reasons, we really want to study aggregates one by one. So the challenges studying these aggregates: they're present at low concentration and abundance, typically only about 1% of the aggregates. 1% of the total protein is aggregates. Typically, the concentration in cerebrospinal fluid is in picomolar level for the aggregates. Like I've shown you, they're very heterogeneous in size and structure. So we have to bridge this concentration gap, going from picomolars to micromolar. Lots of the cellular experiments people do at micromolar concentrations, while typically the physiological concentrations picomolar. Secondly, there's a reality gap. Most of these proteins undergo significant post-translational modifications. They get truncated, they get phosphorylated, and typically we tend to study these proteins in the test tube. And lastly, there's a time gap. Typically, the disease takes decades to develop, and experiments are done in days. And so to deal with the time gap, we develop physical models that we can extrapolate to long times. And to deal with the reality gap and the concentration gap, we've developed methods that we can apply to the real aggregates that form in humans. So what my group have tried to do is develop quantitative methods to detect and characterize individuals, so we can look at the variation, unlabeled, so we can look at the real aggregates in humans, to understand the molecular mechanism of neurogenerative disease. And the types of things that we can measure: the number of these aggregates, their size using the super resolution method, something about their structure, certainly the accessibility of different antibodies, something about their toxicity, something about their ability to be degraded, and something about their capability to seed aggregation. And because we can work with unlabeled aggregates, we can apply this to cerebrospinal fluid and serum and post-mortem brain samples. So it's very widely applicable. And this is the experiment that we did early on which enabled us to realize that we could actually do these types of experiments. We took cerebrospinal fluid from a patient unfortunate enough to have Parkinson's disease, and put it on a cover slip of one of our sensitive microscopes and added a dye thioflavin T. So thioflavin T is non-planar in solution, but when it sees the beta sheets that are present in some of these protein aggregates, it can intercalate and become highly fluorescent. And we looked with our camera, we saw these small diffraction limited spots. Each of these spots we think is an aggregate present in the CSF of the patient. This raised a lot of questions. What are these aggregates? Are they alpha-synuclein? How do these aggregates change during the development of the disease? But it led to the realization that we could actually study the real aggregates presence in humans. So this is an example of some of our assays. If we put the aggregates on a surface and add a proteinase K, then you can watch the aggregates disappear. Oops, that's not playing, but this is the signal you'll see as it disappeared. If you add monomer, you can see them grow. And this is an example of prion protein, which we've added monomer, and we can watch it grow on the surface, and then hopefully you can see it can break into two aggregates, which is a direct visualization of replication of prions in the test tube. We can also use these super resolution images to look at tau. So this is the diffraction limited image of the microtube of the tau in a cell. And this is the super resolution image. And hopefully you can appreciate, we can see much higher resolution. And we can follow the aggregation of tau in cells. We can add a C to a hex cell. Initially, there's very low levels of aggregates, and we can watch the evolution of these aggregates over time. And you can see that we get more aggregates and they get bigger over time. So we can learn about how tau seeding takes place in cells. And what I'm going to focus on now for the rest of the talk is examples of applying these methods to CSF and serum. And these are the references if you're interested in the work. Right, so what we did is take CSF from 10 patients, control patients, 70-year-old from Gothenburg, 10 patients with MCI, mild cognitively impaired patients just at the early stages of Alzheimer's disease, and 10 patients with Alzheimer's disease. We put them into two of our assays. Our first assay, we array thousands of liposomes on the surface. If the aggregate permealizes the lipid membrane, calcium from the bath enters the liposome, and we have a calcium-sensitive dye in the liposome, and the liposome lights up, and we can measure the extent of the fluorescence, and hence measure the capability of the aggregates in the CSF to permeate a membrane. And this is what we found. This level in the control CSF, this higher double amount of permealization in the mild cognitively impaired patients, and the same level as the controls in patients with Alzheimer's disease. In contrast, when we added CSF to BV2 microglial and measured the amount of TNF-alpha they produce, the control and MCI level was this, and we saw a much higher level of TNF-alpha production from the Alzheimer's disease CSF. So it appears the mechanism of toxicity changes during the development of the disease, and inflammatory aggregates form during the development of Alzheimer's disease. What we did to confirm that this is induced by aggregates is add antibodies to A-beta. When we added the N-terminally binding antibody to A-beta, we saw a reduction in the amount of TNF-alpha produced, but not the C-terminally binding antibody, suggesting that these aggregates are fibrillar. In contrast with the calcium influx, we found the N and C-terminally binding antibodies produced a similar effect. They were both effective at reducing the amount of calcium influx. When we looked at the TNF-alpha production and added an antagonist to receptor toll-like receptor 4, RSLA or TAC242, we saw a reduction in the amount of TNF-alpha produced. So this showed that the aggregates that induce inflammation are fibrillar because there's no C-terminally binding by the antibody, and the inflammation is mediated by toll-like receptor 4. To super resolve the aggregates, we use an aptamer that's been developed. It's a short length of DNA, 20 to 100 base pairs, that has been evolved to recognize alpha-synuclein aggregates, but it also binds to A-beta aggregates. So what we did is take this aptamer, extend the sequence with a docking strand, put the CSF on the surface, the aptamer binds the aggregates on the surface, and then we add an imaging strand that binds and unbinds from the docking strand on the aptamer bound to the aggregates. And when it's bound to the aptamer, we can super resolve it. And so this is a diffraction limited image of an oligomer, and this is a super resolution image of the oligomer. And this is a 100 nanometer scale bar, and this is a fibril. So hopefully you can appreciate we can get super resolution images of these aggregates, and again for AB to a super resolution of an oligomer and a fibril. So when we did this experiment, we looked at the numbers of aggregates imaged with an aptamer, and we found no difference between the control MCI and Alzheimer's disease CSF. But when we looked at the size, we saw clear differences. So this is the cumulative frequency plot. Along here we have the length of the aggregate, and up here we have the fraction. So if it goes up very steeply, we have a high fraction of very small aggregates. If it goes up more slowly, we have a higher fraction of larger aggregates. So the aggregates range in size from 30 nanometers, which is our limit, all the way up to 200 nanometers. And you can see by eye, the Alzheimer's disease CSF has a higher proportion of longer aggregates and goes up more slowly. To confirm this, we did atomic force microscopy on these aggregates. So you absorb them onto a mica surface. If we look at these images, control MCI and Alzheimer's disease, as it looks brighter, the aggregate sits higher above the surface. You can just see by eye, there's clear differences. Very small aggregates that don't come up very high in the control, larger aggregates that come up higher in the MCI, and larger aggregates that come up even higher in the Alzheimer's disease sample. If you come down to the zoom here, we have all these small aggregates, less than 50 nanometers in the control, these larger aggregates that come up high above the mica in the MCI, and even longer aggregates that come up higher in the AD-CSF. So quantifying this in the AD, we have things that sit half a nanometer above the surface. These are monomers, and then protofilaments that sit a couple of 2 nanometers above the surface. If we look at the length, we see a clear increase in the length from the control to the MCI to the Alzheimer's disease. So this was interesting because there isn't a crystal structure of toll-like receptor 4 binding to A-beta fibrils, but there is a crystal structure of toll-like receptor 3 binding double-stranded DNA, and it binds hand over hand. The gap, the double-stranded RNA is 2 nanometers in size. So we imagine toll-like receptor 4 binds to these protofilaments that are 2 nanometers in diameter, and it's multivalent. We have multiple toll-like receptor 4s binding all the way along the 100, 150 nanometer length of the A-beta protofilament. And this is why these A-beta protofilaments are so effective at causing inflammation. We're trying to study this process in real time. We have a sheet of light that comes in from the side. We have aggregates that are delivered by the pipette. And then we're going to image the formation of a signaling complex called the midazome, which we're going to tag with green fluorescent protein. So the signaling complex has 6 MyD88 that are tagged up with GFP, and it forms with IRAK4 and IRAK2. And so when we deliver the aggregates to the surface of the cell, you can see these green puncti form, which is formation of the midazomes in real time. And we're trying to see if these larger protofilaments cause signaling by a different pathway, a different mechanism. The process is different from causing signaling with LPS. So to summarize quite a lot of work, what I've shown you is you start with monomer. The monomer forms oligomers, which are good at permealizing the membrane and can lead to dysregulation of calcium homeostasis. And then they can form protofibrils and protofilaments, and these are good at causing inflammation. What we also find, and I haven't had time to show you this, is that in control cells, when we look at the media above the control cells and look at the cell lysate, aggregates form in the controls that are removed as fast as they form. And what we imagine is more larger aggregates are formed as disease progresses. They get bigger, as I show you here, and they become inflammatory and start a positive cycle of aggregation causing inflammation, and inflammation in the glial, in the astrocytes and microglial, causing increased aggregation in the neurons. We find that we think these large aggregates bigger than 100 nanometers are present in biofluids. I'll show you that this data and maybe potential biomarkers. And we're now studying how these multivalent aggregates cause inflammation. So this is an example of using our techniques to image the aggregates. These are alpha-synuclein aggregates in Parkinson's disease serum and CSF. This is a 100 nanometer scale bar. This is in the CSF, and this is in the serum. And what we find in the serum, that the aggregates tend to be bigger. They're a couple of 100 nanometers in size. This is bigger than a coronavirus. And there's a much larger number of these aggregates in serum, about 100-fold more in serum than CSF. And what we find is there are higher proportion of bigger aggregates, alpha-synuclein aggregates in patients with Parkinson's disease than in controls. So this is the data here. So what we did is take the ratio of the alpha-synuclein aggregates to the A-beta aggregates in serum. What we found is all the patients who are healthy have a ratio down here, and all the patients who have Parkinson's disease sit here. So we get very nice discrimination between Parkinson's disease patients and healthy controls. And this is one of the receiver operator curves, where you plot the sensitivity along the y-axis, the 1 minus the specificity along the x-axis. In an ideal case, you'd want a test that went up like this, went up along the y-axis, and then was horizontal at 1, and we're quite close to that. We have an area under the curve of 98%, suggesting this might be a very promising diagnostic for Parkinson's disease. So to summarize, I've shown you that we're imaging aggregates from humans. I've shown you that inflammation plays a key role in Alzheimer's disease and occurs by receptor toll-like receptor 4. I've shown that large aggregates are present in the CSF and serum, and that secretion of aggregates from the cell into the intercellular space and into the blood might be a potential route for aggregate removal. There are detectable changes in aggregate size and aggregate ratio between PD and healthy control, which is a potential for early Parkinson's disease diagnosis, and we need to validate this on a larger sample. And we're currently working at automating this assay and extending it to the Simoa platform. I'd lastly like to acknowledge all the co-workers, particularly Eugenia Lobovanova, Daria Emin, and Yu Zhang, who've been pioneering this work. Simone Ruggieri, who's done all the beautiful AFM imaging of the aggregates, and our collaborators, Henrik Zetterberg and Caroline Williamson-Gray, and I'll happily answer any questions that you have. Thank you.
Hi, thank you so much for your great talk. You're already getting questions, but oh no, we had a raised hand. So let's.
I just promoted Sudipta to the panel.
Oh, you just promoted it.
Yeah.
I
He's
can ask my question during this time. So for the unlabeled monitoring of the proteins, you use theoflavin T, right?
Sorry?
Like for like visualizing in the case of the unlabeled, like your CSF studies and serum studies, do you use theoflavin T, right? Dye, right?
So the selectivity comes from the aptamer antibody, and then the dye is attached so it's visualizing.
Oh, I get you. Yeah. Okay. Yeah. That's why, because it's like if you would, theoflavin T is kind of cross-reactive.
No. So we wouldn't use thioflavin T. No.
Yeah. Thank you. So Sudeepa, you can ask your question.
Thank you so much, Magda. Very nice lecture, Professor Klenerman. I had just one question. Do you see any difference between the sizes of the small aggregates, small oligomers, between the CSF and the sera? What you talked about are things which are 100 nanometers or larger. But I'm talking about a distribution between, let's say, dimers, trimers, and hexamers, pentamers.
So we're not very good at that length scale. So we can only basically see anything bigger than 30 nanometers. So we're not, we wouldn't be in a position to detect that in our measurements. I mean, what we see, like I showed, is most of the aggregates are less than 50 nanometers. The changes are in the tail, where there's a higher proportion of larger aggregates in the disease.
Okay, thank you.
We have a question in Q&A from Marcus Zweigstetter. How specific is the TLR for inflammatory cascade versus, for example, TLR3?
So we've done lots of studies, and we found that TLR4 is the key receptor. The receptor is TLR2, but the response from TLR2 was about 5-fold less than from TLR4. So we think TLR4 is the key receptor to the AB synuclein aggregates, and also recently we've shown tau aggregates as well. So we see it as a key receptor, and we see this as a potential strategy to block this positive cycle by stopping the inflammation induced by the aggregates and stopping the inflammation inducing further aggregation.
Thank you. We have a question from the chat. Ashutosh, very nice talk. Does the size and shape of aggregate correlate with toxicity?
Well, that's what I tried to show. So we think the globular round aggregates are good at permeabilizing the membrane, while the more fibrillar aggregates are much better at causing inflammation. So yes.
And then you have a question from Carmelo. Carmelo, why don't you ask your question?
Okay. Good talk, David. Nice technique. I have just a question for you. In your opinion, is your technique is able to detect the interaction between neurodegenerative proteins and the free lipids on phospholipid on the membrane?
I think we could certainly look at aggregates and ask, is there any phospholipid incorporated in the aggregates? And we've certainly done experiments where we visualize the aggregates binding to the cell membrane. So both of those are possible, yes. It depends what question you want to ask. But yes, we certainly looked at A-beta aggregates binding to membranes. And we certainly could ask, do lipids bind to the aggregates with suitable probes? So yes, in principle.
Okay, thank you.
Wang, can you ask your question?
I think Bin had raised hand earlier than me. So Bin, you can ask.
Oh, thank you. Yes. Hi, Dr. Klenerman. Great talk. I was wondering the potential of your assay for diagnosis purpose. The sample used is CSF and serum. Do you have to manipulate those first before you try to detect them, like enrichment or like antibody trapping?
No, I mean, we actually dilute the serum to do our experiments. For our experiments, but it's basically serum diluted in PBS. We only need 10 microliters. It goes onto our slide and we image. So we don't need very much sample, and we do nothing other than dilute it.
But do you have to, for statistical purpose, do you have to image a large area to get enough average signal?
Yeah. So once it's on the slide, we'll typically image 9 lots of 100 micron by 100 micron area to count enough aggregates. But the method is very, very simple. We've also taken saliva from people and measured aggregates in them. So it's very sensitive, and we can certainly dilute the serum samples. It's not a problem.
How about MCI versus AD?
So we've now refined our methods so we can actually look at A-beta, synuclein, and tau. The problem with the aptamer is we're looking at a combination of A-beta and synuclein. But we've actually been focusing much more on serum than CSF because the sample's easier to get hold of. We've actually found the differences are larger. I mean, it's surprising, but the difference is larger in the CSF than it's larger in the serum than the CSF, and there are many more aggregates. So it seems to us much easier to work in serum than CSF. So we haven't gone back to the MCI samples and asked very detailed questions about the aggregates.
Very exciting. I think especially your direction for SMUA, it would be interesting to compare those two methods. There are already some of the SMUA tests available using phospho-tau-based detection.
Yeah, no, so exactly. I mean, we think, I was surprised, but it looks like the aggregates aren't removed. And as you develop the disease, they seem to accumulate. So it's almost a record of your protein aggregation history. That's certainly what our hypothesis. So I think there's a lot of promise there.
Thank you, very exciting.
Okay, I can answer my questions. So first of all, looks, when you quantify, you show your quantitation results. The disease one, the majority, well, I think that each dot represents one aggregate, right? Or fiber, oligomer, whatever. I think majority of them don't change the size. And you have some dots above it. How would you use this, solve this, heterogeneity, whatever you call it, as an indicator for diagnosis or treatment whatsoever?
So I mean, what we found in quite a lot of experiments is the number of aggregates don't change, but there's a higher proportion of larger aggregates. And so this makes some sense. It's not like you suddenly develop the disease. It's just that there's a shift in the tail. So the way you look at the data is set thresholds and ask, I'm just going to count aggregates that are large or big.
Yeah.
Or
so do you see some patients late onset versus early onset show something significant different? Or I don't think you showed the individual patient results. I think each dot is aggregate. Yeah.
No, so we've only done about 40 patients using these methods. It's all very, very early stage.
Okay.
We haven't done late disease versus early disease. At the moment, we're just going to do more samples to validate our biomarker. But in general, what we seem to be seeing is that there's a higher proportion of larger aggregates as you develop the disease. And so we'd expect someone with early onset disease to show the same changes as a person with late onset disease, i.e. there'll be a higher proportion of bigger aggregates. But we haven't done these experiments. I think in total we've maybe done 50 samples or so. So I wouldn't like to extrapolate too far from what we've done. The main points, I think, are the aggregates in the control patients. I think that's surprising. It suggests that you normally produce aggregates, but they don't accumulate because you remove them as fast as they're formed. But as you develop the disease, there's a change in the aggregates, and the change is subtle. It's in the tail of the size distribution. And that's what our data seems to suggest. So it's not a picture of no aggregates, and suddenly as you develop the disease, you get aggregates. It's a picture that you have aggregates, but those aggregates change in size, shape, and structure as you develop the disease. That's what our data seems to suggest.
So another question I have is kind of a surprise for me that the serum has a bigger effect than CSF. What's the explanation of that?
Well, we imagine, we don't have what we think is When we have neurons in a dish, we know that they secrete aggregates. We think in the brain you secrete aggregates, they get rapidly removed from your brain and end up in the blood. We imagine that some of these aggregates get removed in your liver, but the larger aggregates get removed less efficiently and start to accumulate. The other explanation is that the aggregates are actually not formed in the brain, but formed in the periphery. And it's the aggregates in the periphery that get into the brain and drive the disease. And some of the aggregates that we're seeing in the serum are formed elsewhere in the body. And so it's very difficult to distinguish between those two possibilities. But the simplest thing is it comes from the brain. It's very low in the CSF, but it's been rapidly removed and it accumulates in the blood.
Okay, thank you.
Thank you so much. Marcus, you can ask your question.
Yeah, I would first like to thank you for your very nice presentation. So my question was if you would want to comment maybe on the different activity of the oligomers and the fibrils in terms of membrane perturbation and inflammation sort of initiation. So you want to maybe comment on what you find more important for the disease?
We think the inflammation is the key step. We've done other experiments where we've added pro-inflammatory cytokines to neurons, and we've seen a big increase in the aggregation. So we imagine the inflammation, the calcium disruption caused by the aggregates is early stage, but not too significant. But the key step is once you produce inflammatory aggregates, the microglia and astrocytes produce pro-inflammatory cytokines. Those cytokines lead to increased aggregation, and then you're into this positive cycle that there's no way of breaking at the moment. And we've also shown that the inflammation can cause long-term potentiation deficit, which some people believe is a cellular correlate to memory loss. So we imagine this inflammation is what's causing the memory loss that people have who are unfortunate enough to have Alzheimer's disease, and the inflammation is the key step. If you can get rid of the aggregates before they undergo this slow structural conversion, then you can hope, or you can just reduce the number, reduce the inflammation, then I think you have a potential therapy. So in our hands, the inflammation is the key step because it's so sensitive. You just need very small numbers of these protein aggregates that your innate immune system recognize as non-self, that they're not normally exposed to, that produces inflammatory response that drives the disease.
Thank you very much.
You have a question in the Q&A from Aphrodite. I try to promote her to panelists, but somehow doesn't work. So I'll read the question for you. Thank you for the nice presentation. How do you know that the aggregates you are detecting are composed only of one protein? They could be heteromeric, for example. And the other question is, how was the aptamer generated selected?
So the answer to the first question, it's very interesting. We've previously shown that A beta and alpha synuclein in the test tube can co-aggregate, and we're going to look, at the moment, looking, are they pure, are the aggregates we're detecting pure A beta and alpha synuclein aggregates, or are they co-aggregates of A beta and alpha synuclein, or the A beta pure A beta 42, or they co-aggregates of A beta 1442. Similarly with the tau aggregates, what's their phosphorylation state? No, it's definitely a very interesting question, and we don't know the answer yet, though we've definitely detected co-aggregates in some of the experiments that we've done. And what was the other question?
How was the aptamer generated selected?
The aptamer is generated by a process called selects, where you basically evolve the DNA sequence to get a very high affinity binder to your target, in this case, an alpha-synuclein fibril, and that's been published by a Japanese group who recently used that aptamer.
Yeah, thank you so much. There is a question again. I try to promote this participant to the panel. Doesn't? Oh, he's being promoted. So, where did it go? Where did you do? Oh, yeah. Please, Elton, ask your question.
Okay, thank you so much, David. Very good talk. I wonder about the essays that you did in serum and the question that was raised earlier on regarding the origin of alpha-synuclein aggregates, whether from the periphery or from the brain. It is known also that alpha-synuclein exosomes are also used as a marker, or at least people try. But those exosomes might include, other than alpha-synuclein, also other factors that are clearly or not coming from the brain. Did you think of trying to employ your method on exosomes and try to see the molecular origins of things other than alpha-synuclein embedded in the exosomes?
I think that's a good suggestion. So thank you. No, we haven't done anything like this. We are trying to get hold of an antibody to tau that's CNS specific that would certainly address the question for tau. But for alpha-synuclein, I think what you suggested is very interesting and something that we should definitely think about. So thank you for that suggestion.
Of course, yeah, it's really important because the background of alpha-synuclein in the blood is so high that it's almost impossible to tell anything just from aggregates, I think. Yeah. Thank you.
Thank you.